So it was brought to my attention, someone asked, and I said, sure, we can do this. Could I do with the Krebs cycle and the electron transport chain? Basically what I did on Monday, where I drew on the board, like, what do you actually really and truly need to know on those things? Okay. So here's what we did, what I wrote on Monday: glucose 6-phosphate, the two enzymes that get us to here, hexokinase and PFK. We end up with pyruvate. Okay. Pyruvate can be made into lactic acid. We'll talk more about that today. Pyruvate can also go into the mitochondria, and through an enzyme called pyruvate dehydrogenase, or PDH, can get converted into acetyl coenzyme A. And then that acetyl group gets pulled off, goes into the Krebs cycle, spins around and around. We make an ATP, right? We make 3 NADH, and I forgot to write on here, we also make 1 FADH. And the electron carriers donate their electrons on the electron transport chain, right? And that makes a shit ton of energy. Okay? So basically, this is what I want you to know. I do want you to know that there are cytochromes in the electron transport chain, that oxygen is the final electron acceptor, but this is basically the whole thing. Okay? This is the whole thing. You don't have to know sort of all the different steps in the Krebs cycle. You don't have to know all the steps. What we're going to spend time today talking about is we're actually going to get acetyl-CoA from a different place that can also then go into the Krebs cycle. Okay? We're going to have fat, and we can metabolize fat basically down to here, and it can go in as well. And then we'll talk about primarily which of these steps are the things that control how fast the pathways go, and why in some instances, right now, while y'all are sitting there, you're going to metabolize more fat than carbs, like how we push that around. While when you're doing exercise and the intensity goes up, it becomes more carbs and less fat. Okay? This is the part, okay, where I really, really need questions so that Ty and I can understand what you guys are getting and aren't getting. Okay? So, is there any part of this that seems confusing so far? No? Okay. All right. Good deal. So, I'm going to put the screen down. We'll talk about things. We'll bring this back up in a minute. Okay? And away we shall go. Is it going to get turned on? Hi. Let's look underneath and see if this thing, if there's stuff that needs to get plugged back in or something. Yeah, see, that thing is. Nope, that didn't fix that. I'm going to pull. Cool, cool. Fun times, guys. Can't get that one back in either.
No, like there's something like blocking me out of it. Like, oh my god. What the hell?
How many college degrees does it take to figure out how to plug in? Ty and I have a few between us. Okay, wait, is it working?
That on there?
I don't know. Does that open? You can open that. Nope, of course not. All right, that's fucking cool.
Oh my god.
All right, it's on. Okay, okay. We got this. We're fine. The video's gonna be great. It's just Ty and I rummaging around back here, cursing. Oh my goodness. All right. So while this is warming up and we're getting ready, we're going to start off by talking about fat metabolism today. Okay? Fat metabolism only occurs in aerobic metabolism. Fat does not go through glycolysis. Okay? Does not go through glycolysis. So because of that, we have this giant store of energy in fat, and per any molecule of fat, we have a lot more energy available to us than we do in carbohydrates. Okay. It goes through aerobic metabolism, so it does not make anything like lactic acid that inhibits anything. So it's I don't want to call it a clean form of energy. It's a type of energy where, well, no, it's not a type of energy. It's a substrate where we can get a lot of energy without any byproducts that will cause fatigue. Okay. But because it goes through aerobic metabolism, and because fat is a giant molecule in general, triglycerides are, then it's relatively slow. Okay. But the idea behind this is we're going to get fat from our bloodstream. The fat gets in our bloodstream either from what you've eaten, or it can be broken down from adipose tissue and then dumped into the bloodstream. And once that is done, it gets into the blood, into the muscle. Then it has to go through the cytoplasm of the muscle fiber, and it has to get moved into the mitochondria. Okay. Now, and I don't remember how many of the steps on that entire process that I put on your slides, but collectively, that process is very often referred to as something called beta oxidation. Beta oxidation of fat. There's like 17 steps that go into all of this, and you take the fat and you activate it and you bind it onto something and you cross one membrane, and you take the thing off and you activate it again and you cross another membrane. All of those little individual steps, I may have put those on the slides, but you're not going to be responsible for all of those. Okay. It's going to be a very, very general, the fat goes in, and then we're going to pull off 2 carbons at a time and go into the Krebs cycle. And we're up. Maybe. It's thinking about it. Boom. Okay. So this is the kind of general overview of, right? If you think here is glycolysis, here's the acetyl-coenzyme A that we're going to get at the back end of pyruvate. Here's the Krebs cycle. You've got carbohydrates from glycogen that are going into here. Okay. Generally, we're going to have these fatty acids that come out of adipose tissue and go into acetyl-CoA. And then also generally we've got protein, which gets broken down into amino acids. Sometimes they go in directly. Oftentimes they're going to get converted and go into a carb and go back up in here into glycolysis. Okay. But I like this particular graph. You don't have to know all of the individual steps, but I like that it shows that everything and all of these substrates and all of these different pathways are all working simultaneously. Okay. So your metabolic rate right now, as you sit there, requires some amount of ATP. Okay. And as long as you get that ATP from somewhere, from one of these substrates, from one of these sort of individual pathways in here, we're good. Okay. And if you eat more of one thing, it shifts you in this direction. If you eat more of something else, it shifts you in a different direction. If you haven't eaten, it might shift you in a third direction. Okay. And you exercise and the metabolic rate and demand goes up, then we've got to shift again. But All of this is sort of going on at the same time. Okay. All right. So here was sort of the overview of the things that we were getting out. I'm not going to ask you a specific question about how many ATPs do we get from here and there, other than the 3 versus 2 in glycolysis. That's the only thing that I'm going to ask. Okay. You don't have to know that you make, right, 2 ATP for each glucose molecule that manages its way through the Krebs cycle or any of those things. Okay. But I like to, again, show the math so that people know. All right. As it relates to fat metabolism, we need to discuss kind of how we store fat, what fat looks like a little bit before we jump into things. So the kind of main ways that you see fats in your system primarily are as a triglyceride, okay, as phospholipids, and then as cholesterol, okay. Primarily, like the fat in your adipose tissue is triglyceride. A lot of what you eat is triglyceride, okay? And so a triglyceride is basically a molecule of glycerol and these 3 big long free fatty acid chains hooked together, okay? Anybody seen the Matrix movies? Right? In the Matrix movies, they're the little like sentinel things, look like an octopus that like float around, and they have their long like tentacle things. That's what I don't know why. That is what I always think of when I think of a triglyceride. Is it's a glycerol thing, and then there's these free fatty acids like a jellyfish or an octopus hanging off of there. Okay, so this is as triglyceride. This is kind of our large energy depot that we have in our body. Most of what we eat is going to come out as all of this. Phospholipids are generally kind of neutral fat. This is what the stuff that makes up the membranes of your cells. Okay. Now, in a pinch, you could metabolize this, but generally we don't want to do that. We don't do that very often. And then there's cholesterol, right? Which is a common thing that's going to be in food. That cholesterol typically gets transported around in your blood, bound up on sort of some binding proteins. So you can have HDL, LDL, VLDL cholesterol. Which kind of the predictiveness of that for cardiovascular disease and things and your levels is beyond the scope of our class. But that's kind of the general things that we're going to get. So we're going to focus primarily on what happens to triglycerides. Okay. And so this picture comes out of your book. Okay. And depending upon what book you read, depending upon how things are labeled, all right, some books, especially undergraduate books, call the entire process of breaking a triglyceride down, putting it into the Krebs cycle, and deriving energy from it as beta oxidation. Okay, as beta oxidation. If you read graduate level exercise physiology textbooks or biochemistry textbooks, they will say, well, it's free fatty acid or it's triglyceride metabolism, and beta oxidation is actually like the last step in that process. Okay. So I will, because the book talks about it in that way, oftentimes refer to the global breakdown of fat into energy as beta oxidation. Okay. But if you look in this, even though in the book it calls this beta oxidation, technically the beta oxidation step is the last step. As we bring everything in, we move it around, it gets into the mitochondria, and then the beta oxidation is where we peel 2 carbons off, okay, and stick it into the Krebs cycle. But that's basically what happens. If you've got a free fatty acid molecule, and depending upon what it is, there might be 14 or 16 or 18 carbons in a chain on one of those free fatty acid tails. And at its most basic, those things exist in your blood. You move them into your cells. You break the free fatty acid chains off of the glycerol. So you now have these big, long carbohydrate chains. So just think of that as like everybody in your row, okay? And then when we work our way and to get ready to go into the Krebs cycle, we just take 2 of you and peel you off and you go into the Krebs cycle. Then we take the next two of you and we peel you guys off, and it goes into the Krebs cycle, and so on until we've broken down that entire big long molecule. Okay? That's why fat has so much energy potential, is because it has lots and lots of carbons in it. Okay? Glucose has 6. It has 6. It can basically get broken down and ends up going through the Krebs cycle twice. There's some free fatty acid molecules that per tail have 18. They go through not twice, but 9 times. And so that's why there's this ginormous potential to generate energy from fat. Okay. And because it all goes through the Krebs cycle on the electron transport chain, we don't make hydrogen, we don't cause fatigue in that particular way. Okay. All right. Does that make sense, guys? Does that make sense? Nods of heads. Ma'am.
Our body, as far as energy treatments, like why don't we use this more?
Is it slow? And so if you eat more fat, you will. If you just eat less of everything in general, you will. We are, and this is probably not a thing we should get into today, we are a wonderfully designed energy storage machine as humans. It makes us in some ways different from other animals. Our entire sort of existence, we think we evolved from hunter-gatherer. And so our physiology is such that if we don't get food today, we're okay. And then, but one of our adaptations to that, it seems like, is you don't get food today, but you get extra food tomorrow. We're really, really good at taking that extra energy that we've eaten and storing it away, because there may be these cycles of, well, I got to hunt or gather today, and I may not find anything, so I still have to have enough energy to go do it again tomorrow and the next day. And for a portion of our population, be very careful about all of this. We don't want to make light of. We have lots of food insecurity in sort of our population still, but for, we'll just say, at least the simple majority of people, we have a lot of available energy now, right? When you're hunting and gathering, you're not just going to Chick-fil-A, or you're not going to Cece's Pizza and getting all that you can eat. Plus, when we were hunting and gathering, we moved around all the time. And so, as we will see when we do some other adaptations, we're really good at both breaking this down, but then whenever there's extra, it's hold on to it, hide it away in that way. Okay, so it's a really good question, but it's kind of complicated. Okay. All right. So, and Ty will get into more of this next week, but it's really fascinating that the percentage of your energy at any point in time that is derived from carbohydrate and from fat, we can calculate relatively easily. All we have to do is have you sit and breathe into our metabolic cart. We measure the expired carbon dioxide. We measure the oxygen that you're using, and we can use the ratio of those two things to figure out what percentage of your energy is coming from fat and from carbs. If you eat a regular mixed American diet, okay, at rest, so at very, very low exercise intensities, or basically at rest, you tend to be about maybe 55-60% of your energy is coming from fat. The other 40% or so is coming from carbs. Now, this is what we're getting out of aerobic metabolism. Okay. You see the fat use in kind of this aqua line. You see the carb use in this blue line. And then the x-axis is percentage of VO2 max. So we're resting, we're walking, we're jogging fast, kind of going all out over here. And what you notice that as exercise intensity increases, as energy demand increases, the percentage of our total that comes from fat goes down, and our percentage of our total that comes from carbohydrate goes up. Okay, it goes up. And so There's some control mechanisms that Ty will talk about a little bit next week that are going to play a role in all of this, but a lot of it just comes down to we need energy and we need it right now. And it's faster and easier for us to break down carbohydrate and to stuff those carbohydrates through glycolysis and into the Krebs cycle than it is to do fat. Okay? There's another graph like this that shows at any given exercise intensity, the longer you go, the greater the percentage of your energy comes from fat. And part of that is we all have lots of stored adipose tissue. But it takes 30 or 45 minutes of exercise for the hormones to get released that lead to the breakdown of that adipose tissue to dump those free fatty acids from your adipose tissue into your blood before we can ever present them to your muscles to even be used. And so if you walk for a long time, as you go over time, carb use drops, fat use goes up a little bit, simply because you're presenting more fat, because it takes a little while for that to happen. Okay, so my dad, who used to run marathons, would always talk about it in the olden days. He said, you got to run 45 minutes before you ever metabolize any fat. And that's not actually true. But at about 40 or 45 minutes, you do begin to see a very distinct kind of increase in the percentage of your aerobic metabolism that can be derived from free fatty acids during exercise. And it takes time to release hormones, to break down the adipose tissue, and then to get those triglycerides into our blood so that we can use them. In many ways, it is advantageous to us. It is advantageous to us to use those fats, but we have them stored away, and it takes some signals to be able to get them broken down. Okay, this is called a crossover point. This point here in this graph, it was at about 35% of VO2 max. Depending on how well trained you are, that may shift around, but there's a point and an exercise intensity where you're 50-50, and then there's a point where as you go higher and higher, you're going to tip it towards carbs. And so this used to be a thing that we studied more so. It's just kind of now become an accepted sort of thing. Okay. But it's just kind of a way that we illustrate how these pathways are all going to work together. Okay. We're going to finish today talking about rate control, or the time control of metabolism. Okay. This is the stuff that's really tricky. All right. This is the stuff that's really tricky. So when I say temporal organization, what I mean is the time sequence or the time organization of metabolism. As I said on day one, all of your metabolic pathways are active all the time. In aerobic metabolism, you're getting some energy from carbohydrates and from fat all the time. Okay? All the time. But as energy demand goes up or down, then the pathway that we are going to get the most energy from is going to shift. Okay? And so that's going to be our temporal organization. Predominant pathway depends on demand and the speed of our metabolic pathways and their ability to meet that demand. Okay? So I'm going to show you guys where the control points are in each pathway. This is why this pathway is fast. This is why this pathway is slower. And then we'll finish at the end and try to put all of this together to understand how these things shift around. If it helps you, this is the way that it helps me the most, is that the pathway that we're going to get the most energy from at any given point in time, it just simply comes down to supply and demand. Okay? It comes down to supply and demand. If you have a 6-year-old who gets hungry and you're at home, she doesn't care sometimes about, I want a meal and that I have an entree and some sides and they complement each other and I get a salad for her. Like, she doesn't care about how the food works together. Her thought is, I am hungry now. Give me whatever you have and I'm going to eat that. And then when the next thing is ready, I'll eat that and then I will eat that. And if you don't do that, then everybody suffers. Okay? And everybody suffers. So That's kind of how the metabolism is. It's about supply and demand. You need more energy now. Okay. Let's get it from the place we can get it now. And we'll worry about 30 seconds from now, 30 seconds from now. All right. So most of our pathways have at least one, if not a couple of kind of key rate control steps. We've talked about this, I feel like ad nauseum, but I'm going to mention it again. If we have to rank them on the speed of the pathways, creatine phosphate's the fastest pathway, glycolysis is the second fastest. And here I'm referring to Krebs and the electron transport chain collectively as oxidative phosphorylation, or you could call it aerobic metabolism. Why? I promise you this will be on the test. Why is the creatine phosphate pathway the fastest?
One step.
One step. Good. Why is glycolysis faster than oxidative phosphate or aerobic metabolism?
Doesn't need oxygen.
It doesn't need oxygen. Okay, that's correct. That's not exactly why it's faster, though. It's the same principle, though, right? Creatine phosphate's the fastest because it's one step. So if glycolysis is faster, what can we infer? There are less steps, right, than here. Okay, the speed is generally determined by the number of reactions, the number of steps that are going to take place. Okay. All right. So we're going to talk about within each of these pathways, though, I can make them, especially with glycolysis and aerobic, I can make them speed up or slow down a little bit, even though they have multiple steps. Okay. So Ty, on the worksheet, right, there's a like a table where you write like what goes in, what comes out of each metabolic pathway, and like what the control step is, the rate control step.
Yes, on the actual metabolism worksheet.
On the actual metabolism worksheet. We're about to go through some. Okay. So if we talk about rate control of creatine phosphate, the either increase or decrease in that pathway is driven entirely by ADP. Okay. If there is a question ever in any class about what stimulates and makes a metabolic pathway work or go faster or move forward, you're always safe by saying ADP. Okay. When ADP levels rise in a cell, it pushes our reaction, right? It pushes our reaction from creatine phosphate into ATP and creatine. Okay. We use this until it's gone. So it is driven by ADP and it's controlled by the creatine kinase enzyme. One step, okay? One step, one enzyme, one input, the ADP. That's it. That's why it's so fast. Okay? That's why it's so fast. Oh, now glycolysis. Okay. Good. Now things are going to get a little bit trickier. They're going to get a little bit more complicated. Okay? Technically, because glycolysis has so many steps, we say there are 4 enzymes, okay, all of which I wrote on our pathway. There are 4 enzymes that play some role in controlling the speed of glycolysis. Okay, I will point out the one that is the true slowest step and the true rate control, but there's 4 enzymes that all matter. The first of those is an enzyme called phosphorylase. Phosphorylase breaks glycogen down into glucose. Okay? You have phosphorylase that breaks down glycogen in your liver, so you dump glucose into the blood. You also have phosphorylase in your muscle cells that breaks down muscle glycogen. Okay? A lot of these enzymes in glycolysis are what we call allosteric enzymes. Anybody remember from chemistry or biochemistry or anything else what an allosteric enzyme is? That seems fair. An allosteric, Abby, you want to try?
It needs something to bind to it.
It can bind things, that's right, but when things bind, what happens? You remember? It changes what? You're really close.
Shape,
Shape, yes, but also
function.
function, yes.
Give it to us.
Speed,
Okay.
okay? Speed. Okay. Allosteric enzymes will have things bind to them. And it makes the enzyme work faster or slower. Okay. And so phosphorylase is an allosteric enzyme. And when phosphorylase comes into contact with calcium or ADP or stress hormones like epinephrine and norepinephrine, all of which go up during exercise, it activates the phosphorylase and leads to the breakdown of more glycogen. Right? The body's response to exercise is break down glycogen, dump a bunch of glucose into the system to make sure we have energy. Okay? So that's phosphorylase. This is how are we going to get the glucose into the system? The true slowest step in glycolysis is that third step, right, where we go from glucose 6 to glucose 1,6 or fructose 1,6. Anyway, but it's phosphofructokinase, okay, or PFK. This is truly the slowest step, okay? It's also an allosteric enzyme. As you guys sit here right now, the PFK in your cells is largely inhibited. You have sufficient ATP, okay? You have sufficient creatine phosphate, it's largely inhibited. As we begin to exercise, ADP especially makes PFK work faster. It makes PFK work faster. So this is, we're going to dump more glucose in. We're going to speed up our slowest step as we're moving that glucose towards pyruvate. Okay. That's going to help our reactions go faster. Now, what I want to point out that I think is really important on the list of things that inhibit PFK is hydrogen. When we make lactic acid, we make hydrogen. So if glycolysis, if we tip a lot of our pyruvate over into lactic acid, we make excess hydrogen, which then functions to inhibit PFK and slow down glycolysis. Okay. So when you have acidity and you do this, the hydrogen actually functions to try to shut down glycolysis. And that can be a problem and can lead to fatigue. Okay. So to look at PFK kind of in relation to things at rest, ATP and creatine phosphate are high. PFK is largely inhibited. So it works, but it's going pretty slow. During heavy exercise, when you have a lot of lactic acid, hydrogen is high. So PFK also gets inhibited. That's why you can only do heavy exercise for a certain amount of time. When you're doing walking or other kind of moderate exercise, especially ADP is going to be up. That activates PFK, makes glycolysis go faster, and so we're in a good spot. Okay, it's really this balance between we want to be active and go, but we don't want to make too much hydrogen. That's going to be part of our control here. Okay. The last two enzymes that kind of control stuff in glycolysis are going to be the two enzymes that control where the pyruvate goes. You have lactate dehydrogenase and pyruvate dehydrogenase. Okay. LDH drives pyruvate to lactate. Okay. It's going to help us then to take those electrons from NADH that we made in glycolysis, free up that carrier so it can keep going. LDH is in and of itself not necessarily an allosteric enzyme. It does exist in different flavors. And one of those flavors is more active, drives the reaction faster than the other. So the type of LDH that we see in type 2 muscle fibers is more active. Okay. The type that we see in type 1 fibers, and then honestly the type that we see in cardiac muscle are much less active. Okay. So fiber type's going to play a little bit of a role here, but LDH is going to control sort of what happens to the pyruvate. So we control glucose that comes in, we control getting that glucose to pyruvate, and then we're going to control what's happening with where the pyruvate goes. So to go into aerobic metabolism, we have pyruvate dehydrogenase. That turns the pyruvate into acetyl-CoA. It is allosteric. It gets activated by ADP and calcium, which go up during exercise. And it's largely inhibited by high levels of free fatty acid and or low glycogen levels. So when you eat a bunch of fat, the reason you metabolize a lot of that fat is that fat inhibits PDH, which then limits pyruvate or carbohydrate metabolism going into the Krebs cycle. So we're just going to use the fat. Okay. Now there are low glycogen levels. We'll talk about that later on in class. At certain points during exercise, if you go for a really long time, you use up all of your glycogen. And so when all the glycogen is gone, you're going to have a different kind of fatigue. You're going to feel really shitty during exercise. And part of that is because it inhibits this enzyme. Okay. Okay. So very, very broadly, okay, rate of glycolysis is controlled by how we get from glucose to pyruvate, and then are we going to drive that pyruvate to lactate, or are we going to drive that pyruvate into the Krebs cycle? Okay, so it's that sort of tipping point and balance under these types of things. Okay, glycolysis or going to lactic acid predominates when pyruvate formation exceeds what can go into the Krebs cycle. Okay. This is the primary issue with everything. Okay. We really only make excessive amounts of lactic acid when there's so much pyruvate being formed at the end of glycolysis that it swamps or prevents our ability, right, to be able to go into Krebs cycle. So imagine all of you in here, right, are molecules of pyruvate. We have one door that you can go out that goes to the Krebs cycle. The other door goes out and goes to lactic acid. If I tell the front row to stand up and try to leave the room, if it's pretty orderly, most of y'all can all go out one door, right? And you're not going to get pissed off and go the other way. If I say 2 rows, we're starting to want to go out the other door. If I say everybody in the room get up and get out of the room as fast as you possibly can, we're going to have to go both directions because we've swamped what can happen in the Krebs cycle. We've swamped what can happen on one side. So we're going to go the other direction. Okay? During exercise, we have situations where pyruvate formation tends to exceed what can go to aerobic metabolism at the onset of exercise, and then during very, very high intensity exercise. Anybody go run yesterday? Anybody run yesterday? Bike? Anybody? Like 3 of y'all? Fernanda, how far did you run? You mind me asking? 12 miles. Okay, this may not be what I wanted to. You may not be a good person. Do you remember what you felt like in the first mile?
During the first mile,
Did you feel better in the second mile than in the first?
I felt about the same until.
Okay. Fernanda's way too trained. Anybody feel worse in your first, like you start exercising and you're going at the exact same pace, but you feel worse at the start and it takes you like 5 or 6 minutes to kind of get in and get going and all of that? Okay. That's because at the onset of exercise, we start dumping pyruvate into the system. We make lactic acid, and the Krebs cycle can actually, as we'll see, can go a little bit faster, but it hasn't had a chance to speed up yet. So we make more lactic acid in the first 3 or 4 minutes of a bout of exercise than we would later on. And that's why it feels more difficult. It's one of the arguments for why warming up is important. Okay. I'm not a huge fan of warming up, but there's some real physiology behind some of it. Okay. So the other instance is it's very, very high intensity. You're going at a high intensity, you're breaking down lots of glycogen, you're pulling lots of glucose in from your blood, you've ramped up PFK, we're just jamming all this glucose in. we're making a ton of pyruvate, and it's just more that can go to the Krebs cycle. So to keep glycolysis going, to keep making energy for our high demands, we just make lactic acid. Okay, so those are the two big things. Those are our two big things. Okay, those are the kind of way that all of this gets balanced out. All right. Now, when we talk about how we control aerobic metabolism, it's, I don't want to say it's less tricky, but it's a little bit less tricky. Okay. The Krebs cycle, I'm going to start with the Krebs cycle in here. The Krebs cycle, its rate is primarily controlled by how fast the electron transport chain can send back the electron carriers. Okay. The Krebs cycle is usually not our problem. We get the pyruvate made into acetyl-CoA, the acetyl-CoA goes in. We get acetyl-CoA coming from fat, it goes in. So Krebs can be increased a little bit by ADP and inorganic phosphate. So exercise speeds it up, okay? High levels of ATP and NADH slow it down, but generally this piece of the pathway is faster and works better than the electron transport chain. So in general, aerobic metabolism, its max rate is controlled by the max rate of the electron transport chain. Okay, so electron ETC can be stimulated by ADP levels, by inorganic phosphate levels, and it's inhibited by ATP. So at rest, right now, you guys are running your electron transport chain at a fraction of what its maximal capacity is. We don't want to make any extra ATP if we don't have to. Okay. As this speeds up, right, you need more oxygen to serve as the final electron acceptor. And that's why as you exercise, your heart rate goes up. That's why as you exercise, your breathing goes up. You are trying to bring in oxygen, get it into your blood, get it to the muscles so that it can function here and let us make ATP. Okay, but this is largely the slowest part of aerobic metabolism is here. We're not going to dig into, is it at cytochrome 2 or is it at cytochrome 3 or is it this? Like, just in general, I want you to know that it's primarily governed by ETC speed. Okay. Okay. Couple of points at the very end. All right. During submaximal exercise, walking, jogging, okay, the rate and function of aerobic metabolism is not limited by oxygen. Okay? It's not limited by oxygen. At maximal exercises, when we get the electron transport chain spun up to its fastest rate, there's a chance that it can be limited by oxygen availability. Okay? There's a chance. It is not always, but it could be. Especially in highly trained people that are capable, that have really good cardiovascular systems and pulmonary systems. It could be. Okay? We'll talk about this a whole bunch when we get to aerobic training. One of the primary adaptations to aerobic training is you get more mitochondria in your muscles. So you have more mitochondria, you have more Krebs cycle, you have more electron transport chain. Okay, so imagine I go from having a door like this, now both sets of doors are aerobic metabolism. So now more pyruvate can go there, more fat can go there without having to make lactic acid. That's how we improve things. And then there's a third door and a fourth. Okay, so that's kind of how all of that stuff is gonna manage and is gonna fit together. Okay, here's this, right? Here's a table. You have to remake this table. You don't have to know that the rate limiting enzyme in the electron transport chain is cytochrome oxidase. Okay, you do not have to know that. You do not have to know that isocitrate dehydrogenase is the rate limiting step in, they're calling it citric acid, but it's the Krebs cycle here. I do want you to know these two. Okay, I do want you to know these two. Krebs is basically limited by electron transport chain activity. Okay, and that's how that's gonna go. You will note ADP stimulates everything. ATP basically inhibits everything. The very, very last little thing, okay, And we'll probably ask a question or two about this on the test in a very general sense, okay? This is showing on the left the percentage of energy that is derived from aerobic and anaerobic energy source. This is a short period of time. This is a very long period of time. And then on the right, this is that same chart, but it's listing events, right? So weightlifting, 100-meter dash, almost exclusively anaerobic. Okay, somewhere in the middle, 200-meter swimming sucks. 800-meter dash also sucks. Okay, a mile run here, about 70-30. Okay, 2-mile run, 10K marathon way down here, almost exclusively aerobic. Okay, so just there's this continuum of these things that are gonna be on there. Okay. All right. You guys have a lovely OU Texas weekend. If you go, be safe, making choices. Ty will get with you guys next week on things. And I will try to talk to y'all on a Sunday. All right? See you guys.
Have a good trip.
I'll go